Each cell is a busy and buzzing world. Inside these cells, there are millions of protein factories known as ribosomes. You're probably very familiar with these protein factories. They are responsible for translating the genetic information encoded by messenger RNA into proteins. Each eukaryotic ribosome is a massive complex consisting of 83 individual components, 4 rRNAs, and 79 ribosomal proteins. These proteins play a role in scaffolding the structure of the ribosome. However, is that all they can do? Researchers have discovered that ribosomal proteins do not always assemble into ribosomes. Instead, they can interact with other proteins that are not involved with translation. This ribosome-independent activity of ribosomal proteins can have a significant impact on cell cycle regulation. One key question is, when do ribosomal proteins perform ribosome-independent activities? In order to answer this question, we need to first take a look at ribosome biogenesis. Ribosome biogenesis is the complex process where ribosomes are assembled. It is one of the most crucial cellular processes in a growing cell, as it is a major consumer of cellular resources. For this reason, the integrity of ribosome biogenesis serves as a key checkpoint for cell cycle regulation. This important yet complex cellular process can sometimes be disrupted, which is detrimental for the cell. One phenomenon that disrupts ribosome biogenesis is the imbalance of components that make up a ribosome. During ribosome biogenesis, rRNA and ribosomal proteins are manufactured proportionally. When errors occur during ribosome biogenesis, such as infidelity in rRNA processing, ribosomal proteins may not be able to interact with these defective rRNA and assemble into a ribosome. Thus, the pool of free ribosomal proteins increase. Most unassembled ribosomal proteins are rapidly degraded. However, ribosomal proteins RPL5 and RPL11 combined with 5S rRNA to form a stable complex called 5S ribonucleoprotein particle, 5S RNP. This complex is much more resistant to proteasomal degradation than free ribosomal proteins. This resistance to degradation leads to 5S RNP accumulating and binding to another protein that is not a component of the ribosome, an E3 ubiquitin ligase named MDM2. The formation of this complex inhibits MDM2's ligase activity. This interaction is significant for the fate of the cell because MDM2 is the main inhibitor of tumor suppressor P53. P53 is the key regulator of the cellular stress response network. This protein is so important that it has its own nickname, the guardian of the genome. When 5S RNP inhibits MDM2, P53 cannot be marked for degradation, therefore it is no longer downregulated. This activation of P53 triggers cellular pathways involved in the repair of cellular damage, cell cycle arrest, senescence, and apoptosis. This mechanism eliminates defective cells and prevents them from replicating and passing on defects. In summary, when the integrity of ribosome biogenesis is compromised, free ribosomal proteins increase, which leads to the accumulation of 5S RNP. 5S RNP inhibits MDM2, causing a decrease of P53 degradation. P53 then signals other signaling networks across the cell to repair genomic defects and halt the proliferation of the cell. This 5S RNP MDM2 P53 pathway links ribosome biogenesis integrity to the fate of the entire cell. Since sustained proliferative signaling is one of the key hallmarks of cancer, this 5S RNP MDM2 P53 pathway may hold potential in the treatment of cancer and other illnesses. Scientists are gradually unveiling the novel extra-ribosomal functions of ribosomal proteins. Considering the sheer quantity of different ribosomal proteins, what we know is still only the tip of the iceberg.